two stuff to upload to YouTube, which shouldn't take that much longer before this one uploads. So anyway, I do apologize. I didn't post your PowerPoints and outlines for today until you guys were already taking the exam. So some of you already printed it out, I know. But I do have this PowerPoint, some of the outlines posted for this week. Okay, so we're just going to go through a little bit today. I know you guys just had your first exam, so you're probably feeling, a little brain dead right now. And we'll pick back up on it next week. Okay? All right. So we are going to the cellular level of organization next. We've covered chapter 1 and chapter 2. So when we look at the cellular level of organization, keep in mind that you need to have in the back of your mind the details you learned about chemistry, because a lot of the ways that the cell membrane interacts with things and its structure depends on some of those chemical groups. So of course things build on each other, and that means you can't really just take a test and forget the previous information. You have to know it because it does kind of compound on the next lecture. So that being said, a lot of people are slightly more familiar with the cell than they are chemistry, not everyone, but it tends to make people, just in general, sometimes feel a little more comfortable, I think, than the chemistry chapter does. So anyway, the concepts that we're going to go over today, we're going to talk about the importance of the plasma membrane mostly, so why it's there, the components of the membrane, and we're also going to get into transport methods of the cell. We may not get into those entirely in a large way today, but just a little bit. And this overlaps with the stuff you did in lab last week as well, very well. So that's good. So first of all, this should be pretty easy. This looks like the cells that you did 2 weeks ago in lab. We use the methylene blue to color your own cheek cells. So we've got the plasma membrane. What's the point in the middle you could see? Nucleus. You can see the nucleus. The organelles were too small to see with their microscopes. And everything between the nucleus and the plasma membrane is considered cytoplasm. Okay? So cytoplasm consists of cytosol. Cytosol is 75 to 90% water with other things dissolved in it. And organelles. Organelles, like we went over in lecture, have a specific structure and function, kind of like your organs do in your body, but they may differ depending on the function of the cell and what type of cell that it is. We will go back over organelles in here. I have an activity for you guys to do because I know we went over the general structure of the organelles in lab, but we'll talk about them in a little bit more detail in here. The nucleus contains your chromosomes, which is chromatin whenever it is uncoiled in a cell that is not undergoing mitosis or cell division. And in there it does have genes, your genetic information that encodes everything about you. So this is basically the DNA in the nucleus. So those are our three major parts of the cell. The plasma membrane, also known as the cell membrane, the cytoplasm, and the nucleus. Anybody know what all these little dots are all over the cells here? Sorry? It's a good guess. They're actually not organelles. These are little bacterial cells. So they probably really did come out of somebody's mouth. So if you did see these whenever you looked in the microscope in lab, you probably had little bitty bacteria, usually little things like lactobacilli that tend to hang around your gum lines. At the end of the day, if you had a night lab, just in my experience, most people tend to have more bacteria in their mouth after they've been all day without brushing their teeth. So anyway. So this is the same picture of the cell you should be familiar with from lab. Again, I'm not big on having you just go through and label the location of each one of these for an exam. We're going to end up talking about each organelle. But before we go into the organelles, today we're going to talk mostly about the plasma membrane. So again, kind of building on what we learned from lab, the plasma membrane is a flexible barrier. It is mobile because the cell can move and contort as it travels through vessels, if it's a blood cell, as it moves around in the body. But it's also a sturdy barrier, and we'll see that it's very sturdy and that it doesn't dissolve on itself despite the watery environment of the body. And it surrounds the cytoplasm of the cell, so all the watery stuff in the organelles. So 4 major functions of the plasma membrane. It physically isolates the inside of the cell from the outside of the cell. Now, I think one thing that we talked about in lab is intracellular versus extracellular fluid. All the stuff inside of the cell, so the cytosol, is considered intracellular fluid. And if we're talking about the fluids on the outside of the cell, that's the extracellular fluid. You'll sometimes see it in the book abbreviated ECF. So extracellular fluid is outside of the cell. So again, we talked about the extracellular fluid being a major contributor to what happens in the inside of the cell because of things like osmosis. It related to the tonicity. Okay. Second, it regulates environmental exchanges. This has to do with transport mechanisms. So the structure of your plasma membrane regulates what can come in and out of the cell, what it allows. The plasma membrane allows the cell to communicate with its environment, and it has several ways of doing that we'll talk about today by its components that are attached to it. And of course, it provides structural support to the cell. Your cell does have cytoskeletal elements that can provide structural support to it that are sitting on the inside, usually, of your plasma membrane, and it's going to provide some shape to the cell. All right, so we're first going to look at the lipid bilayer of the cell. You've got three major types of lipids in the cell. You've got phospholipids, cholesterol, and glycolipids. So we're going to start out by looking at the phospholipids, which is the one that we talked primarily about in the lab. So your phospholipids have this phosphate group in the head, and then they've got these nonpolar fatty acid tails. Okay? So if you remember this, and hopefully I know there are questions about this on the exam you just took, if you look at your phospholipid, the phosphate group in the top part of the molecule makes the head polar, and then these hydrocarbon tails, these fatty acids that are the bottom part of the molecule, make the nonpolar tails. And the nonpolar tails face one another in the plasma membrane, and the phosphate heads face the outside and the inside of the cell. Okay, so you guys should know this. Which side interacts well with water? The polar heads or the fatty acid tails? The polar heads. What do the fatty acid tails do with water? They pretty much repel water, right? Because they are nonpolar. Nonpolar by nature means that it does not interact well with water. Now, we talked about nonpolar meaning there's not a more electronegative atom. It's a covalent bond between atoms, and they share electrons equally. But that makes it not very likely to interact well with water. So therefore, you've got these fatty acid tails that don't interact well with water, and these phosphate heads that do. Since the phosphate heads are facing toward the extracellular fluid and toward the intracellular fluid, they can interact with their environment well. But your cell doesn't dissolve on itself because it does have these fatty acid tails that are nonpolar, making up that inside portion. So going back here, you can see the structure of the plasma membrane. There's your fatty acid tails, phosphate heads, facing the inside and outside of the cell. Of course, there's some other things going on here too that we'll talk about. All right? So one important word here is amphipathic. I'll write it on the board too. But amphipathic means that a molecule has both polar and nonpolar parts. So it has both polar and nonpolar ends. It's an amphipathic molecule, each phospholipid. So because it has the phosphate heads that interact with water and the fatty acid tails that do not, we consider it amphipathic, so a polar and a nonpolar part. And these phospholipids make up about 75% of the lipids in your membrane. So again, we said we have 3 major lipids, but phospholipids are the most. They are about 3/4. What's something else that makes up a lot of your membrane that is not a lipid? Anybody remember? Sorry? Well, that's in your phospholipid, but what aside from lipids is also in a high amount in your plasma membrane? Can you recall? Yes. Proteins, right? Remember we had those proteins embedded? So we'll talk about those in a minute. So proteins are important as well. All right. The second major component that we have in our plasma membrane is cholesterol. And this makes up 20% of membrane lipids. So where have we heard about cholesterol a lot?
Cheerios.
Sorry?
Cheerios.
Cheerios, okay. Removing, basically dietary, right? So your body moves cholesterol around it, okay? Basically for lipids to travel in your bloodstream, they have to attach to proteins because lipids are, again, nonpolar. They don't interact very well with the watery environment of the blood. So therefore you've got good and bad cholesterol that's being transported some. And one of the reasons that you have a lot of cholesterol is for membrane repair. Now, if you remember these steroid type molecules, which cholesterol was their base of, they were about 4 ringed structures, and they had a little hydroxyl group at the end of it. So I could pull back up the chemistry PowerPoint and show you, but I won't. I'll trust you guys kind of recognize this at least. But it's got these 4 ring structures and this hydroxyl group. So this hydroxyl group, notice, is going to be close to the polar heads. See the little, if we look at this 3D image, you've got these 4 rings and then you've got this hydroxyl group in red. So notice it's interacting with the polar heads, and then the steroid type rings down here are with the non-polar tails. So therefore, it's going to be weakly amphipathic. It's not as amphipathic with polar and non-polar parts as a phospholipid, but it does have this tiny OH group that can connect with the phosphate heads here. And then these non-polar steroid rings that can fit very easily here between the kinks in our phospholipid tail. So notice that some phospholipids will have a non-polar tail that has some kinks in it, so it tends to sandwich between them. And here's a little bit better view. It just shows some big spaces where you can see how the cholesterol molecules fit in with the plasma membrane. But here you've got your non-polar part, and making it weakly amphipathic, you have this polar part, this hydroxyl group, that interacts with the heads. So several things that cholesterol, being a weakly amphipathic molecule that sandwiches between phospholipids, so several things that it does. Number 1, it stabilizes the plasma membrane. Over a wide range of temperatures. So cholesterol stabilizes the plasma membrane over a wide range of temperatures. Now it's about 20% of membrane lipids. They actually weigh a little less. They don't take up as much space here in mass in the membrane. So they could actually end up being about 50%, but 20% is pretty average. But when we think about stabilizing it over a wide range of temperatures, notice that they help keep the membrane fluid. They sandwich here between these non-polar tails, So that way those tails don't stack on top of one another. So it prevents the membrane from clustering and stiffening during cooler temperatures. So it keeps it more fluid whenever it's colder. However, it gives it more rigidity, so able to pack on itself a little bit better at room temperature. So it helps stabilize it over a wide range of temperatures based on how it packs itself between those nonpolar tails. Now the other thing that it kind of does related to this is it prevents a lot of water molecules from passing through the membrane. So it prevents water from passing between these nonpolar tails. So one thing I did mention in lab is water can sometimes diffuse small water molecules between these nonpolar tails, but it doesn't do it very efficiently. Again, because there's a nonpolar portion in the inside of this lipid bilayer, which water doesn't interact with. That's kind of the nature of nonpolar. So the inside of this is hydrophobic, we say, whereas these polar heads are hydrophilic. So hydrophilic means water loving on the in and outside. Hydrophobic, think water hating, okay, so avoids water. But this prevents too many water molecules, these cholesterol coming between them, from coming between these nonpolar tails in between phospholipids. So they basically make the membrane more rigid and prevent it from turning into mush. So these help stabilize the membrane, particularly over a wide range of temperatures. So they fill in these little spaces between them. All right, and finally, going back to this slide, so you can see our third type of lipid in the bilayer. This is called a glycolipid, bless you. So based on what we've talked about before, what does glyco sound like? Sorry? This thing is so loud. Glycogen, okay. Glycogen was big chains of what? Glucose. So this has to do with sugars. So glycolipids are basically lipids, fatty acid tails, that have carbohydrates or a sugar residue facing out on the extracellular side of the membrane. So it's important to note that these glycolipids are only facing toward the extracellular space. They don't face toward the inside of the cell. They only face toward the outside. Extracellular space or extracellular fluid. They only face toward the outside of the cell, essentially, is what that means. And these are a very minor part of your lipid part of the membrane. They only make up about 5% of membrane lipids. And again, the nonpolar tails face the inside, but these sugar residues, we call them, these little carbohydrates that face toward the outside, are polar, like most carbohydrates. Now these glycolipids help form something known as the glycocalyx. The glycocalyx is made up of your glycolipids and your glycoproteins. And basically what this is, I'm just going to use my own words here, is the sugary coating on the outside of the plasma membrane. So glycolipids come together with glycoprotein. So what does the glycoprotein sound like it is? Yeah, sugar attached to a protein. So we said proteins are in a high amount in your plasma membrane as well. I'll get to that in just a minute. But the glycolipids and glycoproteins basically just attach to integral membrane proteins and nonpolar tails in your membrane, and they make this sugary coating on the outside of the cells. Now, since I have a whole slide on it, I'll go over what that sugary coating does in a minute, but almost all cells have that. So in just a minute, we'll talk about what that does. But first of all, just kind of introducing membrane proteins. So we said that one of the major things that makes up plasma membranes are the proteins. If it just had your lipids, so just your glycolipids, phospholipids, and your cholesterol molecules, it would still be very stabilized over a range of temperatures. It would be flexible but sturdy. But one of the problems is that it would not have many different properties, many different things that these proteins do for the cell, including structural and being able to communicate with their environment, among two of the bigger things the proteins do. So we're going to learn several of the major types of proteins in the membrane and name all of those. But first of all, let's just kind of classify those. So I don't have to erase my glycocalyx over here, I'm going to write it on this side of the board. But you've got two major types of membrane proteins. You've got what we call integral proteins, and then you've also got what we call peripheral proteins. So integral and peripheral membrane proteins. So let's think about those words. Peripheral, what do you think of when you hear peripheral? Okay, on the side. So if you think about peripheral vision, it's not right here directly in front of me, it's on one side. So peripheral membrane proteins are one-sided. That means that a peripheral protein either faces the outside, kind of like this guy, or the inside, which they don't, well, they do show one peripheral protein facing the inside here. But notice that they're only on one side of the membrane. Integral. So if I said that religion is an integral part of my life, what would that mean? Integral means it is deeply embedded. So integral proteins are deeply embedded in the membrane. Now, some of those integral proteins will be so deeply embedded that notice when you look at 1, 2, 3, these three integral membrane proteins, these orange things, they go from the outside all the way to the inside of the membrane. So some of the integral membrane proteins also qualify as transmembrane proteins. Transmembrane proteins are very important because they span the distance between the outside and the inside, and they're also going to make things like these big transmembrane proteins with pores through them. They're really good ways for your cell to interact with its environment and allow things to come in and out of the cell that otherwise wouldn't be able to squeeze between these little phospholipids. So transmembrane integral membrane proteins span the width of the membrane. They go from the outside to the inside. All right, are we good so far? Questions? Okay. All right, so going back to my glycocalyx, we said that the glycocalyx consists mainly of what? Sugars, right? There's sugary coatings on the outside of your cell. So when you picture a cell, just picture like a little sugar cookie, you know, like a little cookie, and you roll it all in sugar before you stick it on the pan and bake it. So that's how most all of your cells are. Most of the little sugars, so going back here showing you what I showed you about a glycolipid, most of the little sugars are going to be oligosaccharides that make up these glycolipids and glycoproteins. And oligosaccharides are just long carbohydrate chains made of anywhere from 2 to 60 monomers. So oligosaccharides are just long carbohydrate chains that face toward the outside of the cell, made of 2 to 60 monomers. So notice 60 monomers, 60 individual little glucose or sucrose molecules are a whole big chain of sugars that only face toward the outside. And this is from a scanning electron microscope, which blows things up quite a bit larger than what we can see them. But notice here, you can see the glycocalyx. So there's kind of a fuzzy layer on the top of these little microvilli on the cell that are showing a thick sugary coating on there. And so believe it or not, that glycocalyx, that set of sugars on the outside of the cell membranes, do a lot of things for the cell. Probably the most major one that you will hear about is cellular recognition. It allows cells to recognize one another. Can anybody think of a good example in the body of that you might have heard of before? Just an idea in your head. Okay, what about transfusion reactions? Most people don't really know why, but most people are aware that if you have type B blood, you don't want a transfusion of type A, right? And part of the reason is because your cells recognize one another and they recognize foreign invaders. So you're going to have antibodies that are going to recognize your A-type blood cells. If you give A-type blood cells to a type B person, they're going to recognize it as a foreign invader. The same way the cells, if you're a type B blood, should recognize your own B blood cells because of that sugary coating. You can think of that sugary coating in cell recognition as recognizing faces. So after we're in here for a week or two, I'll say, I can recognize the faces if you're a student in my class. So, if you bring 5 of your friends in here because anatomy and physiology class is just so exciting and you just know they really want a big piece of this class, right? They just have no better way to spend their 3 hours. I would recognize that those people are not part of this class, okay? Also can allow cells to adhere to one another. So cells can group together based on this sugary coating. For instance, during blood clotting, you have little platelets that tend to clump together that stop you from bleeding out. People might have some bleeding abnormalities if they have a low platelet count. They also help protect the cell from being digested. Yeah?
What you said with the blood transfusion, does that happen when your bone marrow is made? Or is it a different process?
Bone marrow is where your blood cells are made. So yes, it has more to do with antibodies detecting foreign types of things, but right. Bone marrow cells have to be the same as well because that's essentially where those blood cells are made. So we get into the immune system and talk a little more about that like way at the end of AP too. So that's one of the furthest things you'll do, but it is interesting, so a little more complex. Okay. Digestive cells might have enzymes that break up foreign things. So it can help your cells from being protected here. For instance, this is showing the inside of the intestines. And then another thing that might happen in the intestines, it allows fluid to attract to the surface. So remember, carbohydrates, which this glycocalyx is made of, are polar molecules, right? And what do we know about polar molecules and water? How do you describe the relationship between polar molecules and water? They're water-loving, they're hydrophilic. So if you have a sugary coating of cells, or if you have cells with a sugary coating, they are drawing water to their surface because they're hydrophilic. Now, if this is how the whole inside of your intestines look up close microscopically, that means all your intestinal cells are pulling a layer of water toward them. That makes the inside of your GI tract slippery. Now, you may not have thought about your GI tract being slippery, but guess what? You would notice if the inside of your GI tract were dry. It would make it very hard for things to move through your gastrointestinal tract if there were not water in it. Same thing with your blood. It's going to pull fluid toward it and make those more slippery. So it pulls fluid toward the cells. So this glycocalyx is extremely important to the cells, although a lot of people see it as, just a minor portion of what the cell does, but it is very important physiologically. So that takes care of all of our lipids in the membrane. Questions so far? All right. So the next thing we want to look at are some of the functions of these major membrane proteins. Now, I usually have a little activity that we do, and I may do that next week. I kind of assumed that since you guys had an exam today, everybody's brains would not be just all up on activities right away. Am I right on that? So just time to get some basic information, then go back and critically think a little bit later. But this is just a list from your lecture text with some of your integral membrane proteins. So again, what does it mean to be an integral membrane protein? You are a deeply embedded protein. Here are some of the functions of these embedded proteins. So you've got things called ion channels, carriers, receptors, you've got enzymes, linkers, and cell identity markers. So we're going to go through exactly what each one of these is going to do. Okay. All right. So the first thing we're going to look at are channels. Ion channels are going to be one of the most important types of integral membrane proteins that there are, because these are going to be very largely involved with the rest of the physiology that we talk about throughout the semester. For instance, one of the major groups of information that we talk about in Anatomy and Physiology 1 is the nervous system. We spend like the last, at least 25% of the semester on the nervous system. And to understand how your big nerve cells send electrical signals, you have to understand how ions come in and out of these channels. So first of all, what are ions? Differing protons and electrons, right? So if an atom has gained or lost an electron, it becomes an ion. So it has a differing amount of protons and electrons, right? So ions do not come through the plasma membrane well because they have a charge. And remember, these nonpolar tails don't really like water. The same way they don't really like watery things, they don't like things that are polar or charged. So ions are not allowed to squeeze between phospholipids. If ions want to go in and out of the cell, which they do regularly and is a very normal part of how your cell works, they have to use an ion channel. Now these can be kind of fun. All of these are ion channels, so every single one of these in the different colors that you see here are going to be a transmembrane protein that goes from the outside to the inside of the membrane that allows an ion to come in and out. And usually they are specific for the type of ion that they allow in and out. For instance, this might be a potassium channel. It only opens and closes for potassium. It only allows potassium to come in and out. This one is showing maybe this is a potassium channel too. But they may have a sodium or a calcium channel. So it's going to be usually specific for the type of ion it lets in and out. What this is showing are 4 different types of ion channels based on what makes them open. So I'm going to write the names of them here. The top one, we have a leak channel. The second picture, we have a chemically gated, also known as a ligand-gated ion channel. The third one, we have a mechanically gated channel. And the fourth one, we have a voltage-gated channel. Okay. So once again, looking at these four. A leak channel. What does a leak channel sound like to you? A channel that is leaky. If you have a faucet that is leaky, does it matter when you turn it on and off? It just still drips out water occasionally. It's not very fast usually if it's leaky, okay? It's just gonna be kind of a slow trickle, or it goes the most annoying type of leak. Drip, 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 drip. So it slowly lets things in and out. So that's what a leak channel is. And if you notice here, it's showing it randomly opens and closes. There's not really any particular stimulus that causes it to open and close. It's just leaky. It's random. Lots of cell membranes have leak channels on them. The second one is either called a chemically or a ligand-gated channel. The definition of a ligand is simply just a molecule that binds to another one. A molecule that binds to another molecule. And in the body, of course, that's going to be a chemical. So a chemical that binds something. So that's why you'll hear them called either a ligand-gated channel, which is what they named it in your book, but you'll also hear it called chemically gated sometimes. So notice here the important thing about the difference in this purple channel in this figure versus this leak channel is that there's this little red diamond-shaped chemical coming in and binding to a specific receptor on the channel. So notice the only thing that causes the channel to open and allow things to come into the membrane, into the cell, is the binding of this chemical or this ligand. So that's why it is ligand-gated or chemically gated. Because the channel will only open and close in response to binding of a ligand or a chemical. Does that make sense? All right. Third one is this, red channel. This one will make more sense when we talk about the nervous system, really, but this is a mechanically gated channel. So, whenever nerve cells send signals, they have to start by opening just little channels somewhere. So let's say that you touch me on the hand, okay? Or if you touch your own hand, all right? You're gonna feel your fingers on your hand, on your skin. The reason that your brain senses these fingertips to begin with is because you open an ion channel, and those channels, those chemicals that come into the nerve cells, end up causing a cascade that sends signals to your brain, and you say, I feel something on my hand. Well, what first triggers these channel openings in your hand is not just a chemical, so you don't touch your hand and just automatically create a chemical. You open a mechanically gated channel. So mechanically gated channels open in response to pressure or vibration. So some mechanical stimulus like pressure or vibration first opens this channel. And then finally, oh, I'm sorry. Yeah.
You said it opens in response to vibration.
Pressure. Just some mechanical stimulus. And then finally, the bottom, this yellow one, this is a voltage-gated channel. What does voltage-gated sound like it opens and closes in response to? Electricity, or changes in voltage. So these open and close in response to voltage change. Now that doesn't mean you have to get electrocuted for this to happen. But you'll notice in your nerve cells and other cells in the body, there are constant voltage changes. So they've shown you an example here. The inside of the membrane has a voltage of negative 70 millivolts. And notice when it changes to negative 50 millivolts, this channel opens. So they respond to a change in voltage. All right. So those are our different types of ion channels. What makes them different is how they open and close. All right, the next one. This one is a little bit more easy to understand. This is called a carrier. It may also be noted, you might want to write down, that a carrier protein may also be called a transporter. So carriers and transporters are the same thing. So notice for a transporter or a carrier to get something from one side of the cell to the other, the protein has to change shape. So it's kind of like this. This is how it begins, all right? This is your protein. And I wish I had a beach ball or something. Somebody could throw it to me. If I had, I don't know, I'm going to use this marker. I have this marker and it binds on the outside of the membrane. It can't come through because there's a closing. That channel is closed. So what actually happens whenever something binds to it, it undergoes a conformational change. When the protein changes shape, it allows whatever bound to it, so notice here our purple circle, to come in. So a transporter or a carrier just changes shape. Usually amino acids, which are used to make proteins, very important to the cell, tend to enter the cell this way. And we'll give some specific examples next week of transporters or carriers. All right, now this is a little bit more complex looking. Just kind of as a side note, all of these little arrows labeling this receptor, you do not have to know these, okay? So this kind of gets outside of the scope of what we talk about, but you don't have to necessarily know messenger binding site, transmembrane segment, and tyrosine kinase. But these are basically proteins that are transmembrane. They span the membrane, but notice they don't let anything in or out. They are just a receptor. This is basically a receptor sticking out into the outside of the membrane, the extracellular fluid. When something binds to it, it causes all kinds of changes inside. Now, you could get into, probably if you take cell biology, if you take a biology class for majors, they'll probably talk about how this happens, but in here we're not going to get into the details. One of the things that make these membrane proteins particularly interesting is if you go into cancer biology, because there's lots of cell signals that cause the cell to do things in cancers that they wouldn't otherwise based on these signaling molecules. But the big thing is that something binds to a receptor and causes a change. Probably easiest examples, some hormones do this. Hormones travel through your bloodstream, they bind receptors on certain cells, and then they cause changes inside. We'll just say they somehow cause changes. We won't go into the details. And then just a couple more here. So this is going to be enzymes. Sometimes integral membrane proteins are actually enzymes. So what does an enzyme do? You guys should know that. Speeds up a chemical reaction. So for instance, this could be a little drawn figure of the inside of your small intestine. The inside of your small intestines have an enzyme embedded in the cells embedded in the membranes called lactase. I think I mentioned this in the chemistry chapter. What does lactase do? Break down lactose, okay? So it breaks down lactose so you can digest milk products. People that do not have this integral membrane protein, lactase, are lactose intolerant. They cannot break down lactose. So that's an example. It might be an enzyme just embedded in the plasma membrane. They don't have to be floating around all the time. Cell identity markers, these are actually your glycoproteins. We already talked about that. We said that glycoproteins and glycolipids make up a structure called your what? Calyx. Yeah. So basically you can see that there's these transmembrane domains. They travel through the membrane, and then they have these sugary molecules on the outside. So they just serve as cell identity markers. That's an example of like ABO blood grouping. Cells can identify one another. And then finally, I think these look kind of cool. These are called linkers. Linkers are transmembrane proteins that hold on to other proteins on the inside and outside of the cell. Basically what these do, they provide structural stability to the cell, So linkers will provide structural stability by anchoring things inside or outside of the cell. Okay. So specifically peripheral proteins, because the 6 things I just mentioned were integral membrane proteins. Peripheral proteins that only sit on one side of the membrane do a variety of things. Some of the things that they do, they are going to structure and support the membrane, kind of like what you see here bound with these linkers. They anchor integral proteins in place on one side. They can also move things within the cell. You may have peripheral proteins that are attaching 2 cells to one another. And they can also help alter and change the shape. So these are just things that peripheral proteins or one-sided proteins will do. So just various activities. I didn't make a picture of each one of those. All right, questions so far about proteins in the membrane? Good? All right. That's something I'm going to do next week. So I'm going to skip through these. Go to my next question here. So I posted a little list of critical thinking questions. If you didn't print them off yet, because I did post those while you were taking your exam earlier, that's okay. But here's one of the first questions. So when some cells release ligands, so remember we said a ligand is a molecule that binds another molecule, they are sent through the bloodstream to every area of the body. So your bloodstream might release a hormone that goes all over the place. Why do you think that only certain types of cells will respond to a particular ligand? So I'll give you a minute to read over that and think about the answer. So go ahead and read over that and think about your answer here. Why would only certain types of cells respond to it? Anybody think they have a good answer? Okay, what's your answer?
I figured that because different cells do different things, that we will only allow it to do certain things based on what it's supposed to do for that cell.
Okay, so he said that since different cells do different things, which we know they do, cell specialization, it might only allow certain cells to do certain things. The key here is whenever a ligand is released, it is usually specific to a certain type of cell. So what we want to go back to, the type of cell we looked at here that is related, is a receptor. Notice that receptors might stick out of a cell that are specific for specific ligands. Just anything can't bind this receptor. So this, for instance, might be a kidney cell. And what was released might be something called ADH, antidiuretic hormone. You don't have to know what that is yet. But basically what that does, it binds to kidney cells, and alters their permeability of water. Okay. So only those kidney cells are going to have those ADH receptors on them. So therefore, every hormone or every chemical that is released throughout your body won't bind and react on every single cell. They have to do with receptors. Also, one thing we'll talk about when we go to the endocrine system and we talk all about hormones is your cells are able to upregulate or downregulate receptors. What do you think upregulation means? Make more receptors. So therefore, if your kidney cells make more receptors to antidiuretic hormone, guess what? They're going to be more susceptible to the hormone's effects. They can also downregulate receptors. What does downregulation mean? Make less. We'll talk a little bit next week and a lot going toward exam 3 about downregulation of receptors during diabetes. So we're thinking about things like glucose and insulin. So their down regulation means that your cells respond less to glucose. So we'll get into that when we go to metabolism. Always pretty interesting. All right. So we talked a little bit about this before, but membrane fluidity. So membranes are fluid because of the structure. You've got phospholipids, you've got those integral proteins. Integral proteins are usually like glaciers. They kind of are just like floating through the ocean. So they're not usually just fixed in one spot in the plasma membrane. They can usually float through. So usually those proteins and lipids are very mobile in their own half of the bilayer. So for instance, here's a nice picture of our plasma membrane. These phospholipids can flip around one another and move about 10 to the 7 times per second. So that's like 10 million times a second. Okay? So they move a lot. Because these little phospholipids flip around one another so much in their own half, it makes them very fluid. And the proteins can move as well, whereas cholesterol can kind of stabilize it there. The other thing, we talked about kinks in the fatty acid tails. When we think about the structure, what do kinks in fatty acid tails allow? Or what do they not allow? They don't pack very well, right? So here notice that there are saturated tails. You've got straight fatty acid tails and they're all packed. Here you've got kinks between them. This membrane is going to be more fluid because it has kinks between them. They don't pack. Kind of like oils versus solids at room temperature, right? We talked about that. So that's going to affect your plasma membrane as well. Now, one thing that does not happen a lot are these sides flipping one another. Only about once a month can phospholipids trade places with the inside and the outside. The reason for that is because it's very hard for these phosphate heads to cross through this nonpolar part of the membrane. Okay? So they don't really flip-flop very often, but they constantly can move around in their own half of the bilayer. So those things contribute to the fluidity of the membrane. All right. Next major thing we want to talk about is selective permeability. It's important to understand what that means. Selective permeability simply means some things pass through the membrane easily, while others do not. Some things can go through really easy while others don't. So I'm going to make 3 categories here that we can talk about. So if something's going to pass through the membrane, some things can pass through between phospholipids really easy. Easy, hard, or not at all. So first of all, what we are discussing when I say easy, hard, or not at all is how easy is it for a molecule to squeeze between phospholipids and get in and out of the cell, because remember, this is fluid. Now, of course, the cell membrane can become more permeable by putting these transmembrane proteins in there, these integral membrane proteins. We've got those channels and carriers that will allow large things like macromolecules to pass through, okay? So thinking about this, we said that a macromolecule has to come through the membrane. Macromolecule, like it sounds, is a big molecule. Through like a channel or a carrier, okay? They're not going to be able to come through something small. So not at all, we'll say macromolecules. They will not squeeze between phospholipids at all. They will need, I'm going to say, a carrier or a transporter, whatever you like to call it. So they can only go in and out of the membrane if they attach to a carrier that changes shape. So based on what you think, what types of molecules do you think could squeeze between those phospholipids really easily? Sorry? Okay, she said micromolecules, things that are, first of all, small. Now, they not only need to be small, but molecules that can squeeze between these phospholipids need to be small and nonpolar. Nonpolar means hydrophobic, okay? So you got these fatty acid tails that are going to prevent polar molecules and charged molecules from coming through easily. Now, how that makes sense? This might be a slide to review if the polar and nonpolar still seem a little foreign to you. But when we think about solubility, we're thinking about something dissolving. So something has to be able to pretty much dissolve through that phospholipid bilayer so it can go from the inside to the outside of the cell or vice versa. Polar covalent bonds have more electronegative atoms, so they've got a partial positive and partial negative end. Non-polar covalent bonds mean they share electrons equally, and these are going to be things like fats and oils at room temperature. Okay? They're going to be fatty. They don't interact well with water, so they're hydrophobic. So in general, when you're thinking if something dissolves well or something is soluble, think about like interacts with like. Things that are hydrophilic like each other. Water-loving things like things that dissolve in water. So therefore, ions and polar covalent things tend to stick together. They interact well with the phosphate heads, which are also polar. Hydrophobic things interact well together. So non-polar covalent bonds tend to interact well. So therefore, if we want something to dissolve and pass well through this plasma membrane, because they have a long span of fatty acid tails to cross, generally if they're going to pass through very easily, they also need to be nonpolar or fat soluble. So the things that pass through that membrane the easiest are small nonpolar molecules, or other words, small hydrophobic molecules. Now here we have some examples. Oxygen, CO2, nitrogen, and little steroid hormones. They have like those 4 rings. These make sense because lots of hormones are steroids, and they may need to cross the membrane to cause effects. Oxygen and CO2 are nonpolar molecules. So why would oxygen and CO2 need to be able to come in and out of the membrane easily? Breathing, right? Your cells have to have oxygen to give off CO2. We don't want that to be something hard to come in and out of the cell, so that makes sense. So I had easy, hard, and not at all. Things that are hard to go through the membrane. Small, uncharged polar molecules. So we said that things that come through very easily and fast are small and hydrophobic. Things that are hard are small and hydrophilic, or we'll call that small and polar. So notice if it's going to squeeze between phospholipids at all, like you said, they have to be what you said, micromolecules instead of macro. They need to be small, period. But they're going to come through very fast. If they're non-polar, it could dissolve in these fatty acid tails. They're going to come through very slow if they're water-loving, because it takes them a while to go between these tails. So examples of small, uncharged polar molecules. Water, something called glycerol, that's kind of like the backbone of a fat. Urea, and ethanol. They're just small molecules that are polar. All right, things that do not go through the membrane at all unless they have something like a carrier. Large polar molecules, so macromolecules. The other thing that does not pass through the membrane at all are ions. Now, do you think ions are large? No, but they have a charge. Things with charges do not interact with these fatty acid tails. Hence why we had the whole list of ion channels up here a minute ago. You remember we called those leak channels, chemically gated channels, mechanically and voltage gated channels. They were ion channels. Ions have to have a channel because they cannot interact with these fatty acid tails. All right. So, I have a little activity for you to do. So, take a minute or two. I have 6 molecules at the bottom, and you're going to put them in a category of fast, slow, or no. Are they going to diffuse between phospholipids quickly in the membrane? Are they going to be able to diffuse through the membrane but do it very slowly, or not at all? So take a few minutes. You can talk to the people around you and figure out your answers, and then we'll see what's right. Okay? So fast, slow, or no? So if you're doing it on your own, just chat with somebody else once you think you got the answers to see if they came up with the same thing. Yeah. Well, the reason for that is a good question. Two oxygens bound to one another become non-polar because neither one of those are electronegative connects. And when you have a hydrogen with it, it makes it polar. Acetic acid? Yeah. And that's why it's a critical thinking. Because it's about looking at the molecule here. Okay. Well, and again, it is supposed to be hard. It's critical thinking, so that's why I said we're going to go over it. Right. And you don't have to know each one of them. The point of this is not so you can memorize the structure of estrogen and things like that. No, I'm not saying you are. I'm just saying it's so we can look at these and critically think about them. So we're going to go over them. That's why I just said make an essay. And it doesn't really have to do with the acid. It has to do with the charge. Yeah. That's what it has to do with the charge. Notice things with the charge can't. It doesn't make it non-polar, but it makes it have a charge on it. And things with charges don't have to. Right, the ions just don't have to. It's not a positive, it doesn't have a charge at all. The reason that it takes the hydrogen off is it becomes an ion. So notice this became an ion when it switches from this to this. Okay? So that's the point of this question, is it has an ion. It's an ion here. It just lost the hydrogen. All right. We'll talk about it. That's what we're doing.
Still won't get it.
Come by and talk to me if you still don't get it here. It's just critical. All right. So things that go through fast, slow, or no. I didn't mean to put the answers up here yet. Just want to go through it. All right. So fast. Which ones of these do you think would come through quickly? Oxygen, because it was on there, right? Now, oxygen is a small nonpolar molecule. If you aren't sure how this is nonpolar, when you've got two oxygens together, even though oxygen is a more electronegative atom, they balance one another out because one is not more polar than the other. So it's small and nonpolar. What else is going to go through quickly? Estrogen. Okay. So estrogen, the reason for that is if you notice these four rings, this is a steroid. Okay. So steroids are small hydrophobic molecules. And you may not notice that it's small when looking at it in a picture. That might be a little harder. But you should notice the general structure of a steroid. So this is small, okay? So usually those hormone type things will come through easily. All right, slow. Notice in our hard category, it's hard for them to come through. They'll come through slowly. Are going to be things that are small and polar. What things here do you think are polar molecules? Okay. These are our polar molecules, acetic acid 1.5 and glycerol. Now this might be a little harder to see, but they've got these hydroxyl groups on here. OH, and this is a carboxylic acid, of course. Not hydroxyl, but carboxyl group. But these are polar molecules. So things that are small and polar will come through pretty easily. I had glycerol listed earlier, so that might have made that one a little easier. Now what usually confuses people the most is that it has acetic acid 1.5 and acetic acid 7 on here. Because they're like, it's the same thing. I don't understand why they're different, okay? Notice that when we look at our not at all category, our macromolecules or ions, things that are very large or ions are going to be in the category of they cannot pass through at all without a carrier or an ion channel. So sucrose is a very large molecule. If you compare the bonding structure to these things that came through slowly, it was large. So sucrose is going to be a large molecule. It's a disaccharide that would need a carrier. Yes, it is polar, but it's big. That's why it couldn't just diffuse through the membrane. So it doesn't matter what it is if it's big. And the other thing, anytime you see a picture of something with a charge on it, if it's an ion like a positive or a negative, it is not going to diffuse between phospholipids, okay? It is going to have a channel. Things that are, I'll say ionated, not a word, but ionized, something that has a positive or negative charge will not squeeze between these. So anytime you see that, it would have to have an ion channel. So sucrose and acetic acid pH of 7 would not come through there. Now, would I ever show you something without showing you a molecule and say this at a pH of blah, 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 would it come through? No, because that would be really, really hard for you to see. But the point of this was just so you could see this is a small polar molecule and then compare it to this, but this has a charge. Charges don't come through. Okay? A little bit of critical thinking. And I think since this is an active slide, it came out in your slideshow, I believe. So I believe you have those answers there. Questions? Okay. So